Hi everyone, my name is Nishant Saxena and I'm the product manager for protein purification portfolio at GenScript Inc. USA. Today I'll be talking about novel solutions for high throughput antibody and protein purification using the Magnetic Beads platform. The following are the contents of the presentation. So I will basically be discussing about the current tools which are available in the market and which has been used by a lot of customers, and the need that is basically seen for developing the MAG-SA Plus protein and antibody purification system. I'll be talking about the MAG Plus protein and antibody purification system itself. Then I'll be talking about the menu that we have for magnetic beads for purifying the antibodies, then followed by the magnetic beads that we have for tag protein purification. And finally, I'll be talking about the solutions that is provided by GenScript to reduce the culture volume from several liters to a 50 ml tube so that you can still process your samples inside the MAG-SA system. Now, these are the current tools which are available in the market, and you can see that there is a variety of tools that customers use for their purification needs. And it can range from a very simple and a very inexpensive method of gravity column to a very expensive method like automated liquid handling platforms. Now you see that the sample throughput is pretty variable for all the systems. It can range from 10 samples to 96 samples. Now the time that is also required for processing these samples is pretty variable, and it can range from 3 hours to a maximum of up to 8 hours. So also these systems, they come with their own advantages and disadvantages. So if we talk about these three systems, the chromatography system, the liquid handling platforms, and the extraction instruments, we see that almost all of them, they have a pretty tedious process of preparing the samples, because all of them, they involve the centrifugation step, they involve the filtration step. Also, they involve the concentration step, which takes a really long amount of considerable amount of time. So if you see that, the price of the instruments also is pretty high. So if you see the range of the price, they range from anywhere between 80K to a maximum of up to 200K. Also, the gravity columns, they are inexpensive, but they have other disadvantages, like there is too much labor which is involved. It has low throughput and it has a very low efficiency. Now, keeping this picture in mind, the Amgen and GenScript, they came up with this idea of coming up with a new system which is based on magnetic beads and which can purify your samples very fast without compromising the throughput and without compromising the quality of purity that you get after finishing your purification. So the magnetic beads technology actually is not new, and it has been known for many years, and it has been recognized as a very cost-effective way and an efficient way for purifying proteins and antibodies. Now Amgen and GenScript, they came up with this idea of developing this instrument, and they put this into a concept where we have this MAG-SA instrument and MAG-SA Plus instrument, where it's a walkaway instrument where you just purify your samples in minutes rather than hours. So here is the concept of the purification. It's pretty simple. So once you finish culturing your samples, you just add the magnetic beads to your sample. Your magnetic beads will capture your proteins or antibody of interest. And once that step is complete, the automated instrument, our MAG-SA Plus instrument, is basically going to come into picture, where it's going to perform all the washing and elution steps for you. And once that elution and wash step is complete, your purified protein or antibody of interest is going to be ready for use. So it's as simple as that. So there is no centrifugation, no filtration involved in this whole process. There is no chromatography system because this is a different system. This is based on magnetic beads. There is no column packing because there is no columns involved. It's a very easy to use instrument and pretty automated system where you just set up the protocol and walk away. It has a higher throughput because it can process up to 12 samples at a time, 12 50 ml samples at a time, and up to 60 samples in a day. Now let me talk to you about our MAG-SA Plus purification system, its capabilities, and its key features. So the whole idea of developing the MAG-SA and the SA Plus system was basically to shorten your purification time. And that time basically is involved in several steps, which actually is involved when you are using the column-based system. When you are using a column-based system, you are using centrifugation, you are using filtration. That takes a considerable amount of time. But now when you are using the MAG-SA Plus system, which is based on magnetic beads, you completely eliminate those steps. Now that saves you a lot of time and then saves you a lot of labor. And that is the whole idea of developing the MAG-SA Plus. The entire system is closed to minimize the endotoxin contamination. So in case if you're purifying your antibodies, you don't have to worry because the entire system is closed, and we have a function built in the system to basically minimize the endotoxin levels. And I'll be talking about that in the coming slide. We have complete washing and elution steps, which is performed automatically. So you just come, set up the protocol and walk away, and your instrument is going to take care of the rest. The system is capable of purifying up to 12 samples at a time. The sample volume can range anywhere between 2 ml to 50 ml, and it can process up to 80 milligrams of antibody or protein per sample. Now, these calculations is basically based on two of our magnetic beads, our MAG protein A beads and our nickel charge beads. Now, both these beads have a binding capacity of about 40 milligram per ml. And the maximum amount of beads that you can process inside the MAG-SA system is 2 ml, that is 2 ml settled beads. So if you just multiply 40 times 2, it comes to a figure of 80 milligram. And that's the reason we reached this conclusion that you can process up to 80 milligrams of antibody or protein per sample in the instrument. The typical cycle time for 12 samples is anywhere between 45 to 48 minutes. The instrument is very programmable and very accommodating. It's like a big iPad where you can just touch the screen and set up the protocol. And the minimum elution volume that you can get using the system is 400 microliters. So, and that too in a 50 ml tube. So even if you have hard to express proteins, you can elute it with a very small volume up to 400 microliters, which is going to give you a very concentrated protein. It has a very user-friendly software. As I just said, it has a very big touch screen, and these touch screens can be seen here. All these three are the touch screens. It has various interface like pre-treatment tab, processing tab, and stripping tab. Now, pre-treatment tab is basically used for pre-treating your instrument with NaOH in case if you want to reduce the endotoxin levels. We also have the UV lamp. So you can just start this and it is going to sterilize the chamber where you're planning to process your samples. Also, the processing tab is the main center where you process all your samples. You just have to tell the instrument how much magnetic beads you are using for that particular run, and you have to select the correct ports and the number of cycles that you want to use for that particular run. Once you tell everything, you just click on the start button and a buffer usage amount reminder screen is going to pop up, which is going to tell you that this is the amount of buffers that's going to be required for that complete cycle. Once you go back and check that you have enough buffer in your bottles, you just hit start, and after that, the instrument is going to take care of the rest. The stripping protocol is also included in the instrument. So since our beads actually can be reused, so there's a stripping protocol which is also used in the system. So the instrument basically can regenerate the beads for you, can just prepare the beads for you again once you finish purifying your samples using those beads. So you just purify those beads and then you can reuse them again. There's a USB software, which is a USB basically port, which is present on the instrument that is for updating the software, and that is used for transferring the protocols to your computer. Please note that you cannot attach the computer to this MAG-SA Plus system because the MAG-SA Plus system is self-sufficient. You can save up to 50 protocols on the system, and you can recall them at any point whenever you plan to use them next. Now, this is a slide that basically tells you about the fast workflow for antibody purification, and it just compares the traditional way of purifying your antibodies and and compare the way if you purify your antibodies using the MAG-SA Plus system. So using the traditional system, you see that you have to still centrifuge and filter your samples, perform column packing, then do sample loading, washing, elution, and then finally followed by regeneration of your resin. In the case of MAG-SA Plus, it's a pretty simple and straightforward process. Once you finish incubating your samples with the beads, you just bring it to the instrument. And the instrument is going to perform the wash and the elution steps, and the entire cycle is going to be completed in 45 to 48 minutes. This is a similar slide that actually talks about the workflow of tagged protein purification. So again, comparing the traditional and the MAG-SA Plus system. Here you see that in case if you have the bacterial cells culture, you have to centrifuge your samples, lyse your cells, and once the lysis is done, you still have to centrifuge them, then filter them, and then finally you perform column purification. In the case of MAG-SA Plus, you still have to initially centrifuge and lyse your cells. And once the lysis is complete, you just add magnetic beads to your samples, incubate them for the required amount of time. And once that incubation is complete, you just bring it to the instrument, and the instrument is going to take care of the rest. Now, this is a link that basically tells you about the way the MAG-SA Plus works. So let me just quickly show you how the instrument is going to work. So you can see that how easy it is for you to basically incubate your samples with magnetic beads and then eliminate the need for centrifugation and filtration steps, and then just purify your samples using the MAG-SA system. Now, this is a typical setup for our MAG-SA Plus instrument, and you see here that on the left-hand side, you see the MAG-SA Plus instrument, and on the right-hand side of the instrument, you see different buffer bottles with different port numbers written on them. Now, these port numbers, they basically tell you about the different buffers that you are using. They are also connected to different tubings, and all these tubings are basically labeled with the same port numbers that are written on the bottle. So this port number, you can see here, it's written here, and the same port number is written on the tubing as well. And these tubings are actually then connected to the ports on the instrument. And these ports numbers are also labeled with same labels which are on the tubing and which are on the bottles. So what you basically have to do is that once you fill up these bottles with different buffers, you just close all the lids and then put this tubing on these bottles. And then you just connect this tubing to the instrument here on the ports. And once this is complete, your instrument basically is all set up and ready to use. Now you have a choice of up to 5 buffers to use in the instrument. So you can either use them for elution, or you can either use them for wash depending on your needs. You have 2 buffers, which is basically for washing the nozzles and for wash station. And you have 1 port, which is for dedicated to waste collection. Now, these are the features of the MAG-SA Plus instrument. So here you see that the number of samples that can be processed at one time is 12. It has a minimum elution volume of 400 microliters. So you can elute using 400 microliters in a 50 ml tube. The compatibility of the tubes is 50 ml. The number of buffers that can be used in the purification is 5. So and again, you can either use more wash buffers or more elution buffers depending on your needs. The time required to process the samples is anywhere between 45 to 48 minutes. The range of magnetic beads that you can use inside the MAG-SA Plus system is 100 microliters to 2 ml settle beads. That's the minimum and maximum amount of beads that you can use inside the system. The USB port is basically for updating the software and for transferring the protocol that you have saved on the system. The reagent channels are 7, and there are several ports basically which can be interchanged, and that's basically for wash and elution buffer, and you can save up to 50 protocols on the system. Now, this is the data that basically tells you about the comparative analysis between MAG-SA versus the chromatography system, and the recovery rate that we get using different clones of antibodies. So here you can see that the recovery rate is pretty similar with both MAG-SA and with the chromatography system. But what is difference? What the difference is that there is a lot of labor saving, because there's a lot of time saving, because you're not centrifuging your samples and you're not filtering your sample. So if you're not filtering your sample and if you're not centrifuging your sample, you are saving a lot of labor. Also, you're saving a lot of money on the labor that you are paying that labor for processing those samples. Also, the time itself for processing the samples is very little, and that saves you a lot of time. So it's basically a win-win situation when you're using an MAG-SA system versus a chromatography system. It's a similar slide that basically compares the antibody recovery rate between the two systems, between the MAG-SA system and the automated liquid handling platform. And here you see that with different clones, you again see that the recovery rate is very, very similar. And but what you save on is a lot of time and labor and money. So MAG-SA doesn't compromise on the quality. You still have the same recovery rate, but you actually save a lot of time and you save a lot of labor when you're using the SA or SA Plus system. Now, these are the several MAG-SA advantages, and these are the key steps which are actually involved in the column-based system. So here, as you can see, that there are these portions which are actually written in black. You see that these are the systems which is basically different between the column-based system and in the MAG-SA system. So you have to purge the system and fill it with buffer lines, but in the case of MAG-SA system, you just have to fill them with the buffer lines. It's pretty straightforward. You have to connect and equilibrate the columns in the case of column-based system, whereas in this system, you just wash the beads and add them directly to the samples. Also, you have to concentrate and load the samples in the case of a column-based system, but in the case of MAG-SA system, you just transfer the beads to 50 ml conical tube, and that's it. You are done with it. The one that are actually highlighted in red here shows you the steps which are involved for processing 12 samples, and the number of hours that you will require for purifying the samples when you are using the column-based system. So here you see that the number of, for 12 samples, you need these number of hours for processing these particular steps. So when you add them all, it comes to a total of 9. Also, when you're using a column-based system, there are several reagents that actually comes into picture. You have to use several reagents like disposable filters and columns, which comes at an additional cost. So if you add up everything, you see that you are going to be spending about $1,200 to $3,600 dollars for processing 12 samples. So you can see that this is the cost that's coming up with the column-based system. Now, when you are running an MAG-SA Plus system, there are some costs which are still there, like you still have to fill the buffer lines, you still have to wash the beads and add them to the samples, you still have to collect and elute the protein, and you still have to perform the desalting step. But the whole process is only 2 hours. So you see that the labor cost here is only for 2 hours as compared to 9 hours in the case of the column-based system. Also, the reagent cost is only for the magnetic beads, which is only $5 to $54. And then if you see at the total number for 12 samples, you are looking at the figure of $60 to $648 for processing 12 samples. So you can see the difference in the cost, how major is the difference in the cost if you are using the column-based system versus the MAG-SA Plus system. Now, of course, you you have a big advantage of processing your samples in limited time, because you're processing your samples just in 45 minutes. You have lower cost per sample because our beads can be reused. Our beads can be reused up to 40 times for protein A beads and up to 100 times for our Magnetical beads. So if we are using our reusable beads, you see that you have a huge advantage of using them and cutting the cost even more. Also, you have like several hidden costs and time that are basically time consuming, and those are the steps of centrifugation, filtration, and concentration. Now, why I'm talking about concentration here, it's because our instrument can give you as little as 400 microliters in elution buffer. So if you're eluting with 400 microliters, your sample is going to be very, very concentrated. And you do not have to concentrate your samples, which you are generally performing in case if you are using the column-based system. Also, the simplified and scalable, it has a simplified and scalable workflow, assuring minimal training because it's a very simple system, and anybody can be trained in just one go for the MagSA Plus system. The purification of microliters to liter sample volumes and quantities, so you can actually use our magnetic wand, our magnetic block that I'll be talking about in my slides, and you can process like several micrograms or several milligrams or even up to grams of samples using our MagSA Plus system. So this is a summary of the slide. So you can see that you can save time, that is 45 minutes. You can save labor, that is no centrifugation and no column filtration, and that is equal to your MagSA Plus. Now let me talk to you about our dynamic magnetic beads portfolio. So this is our menu for antibody purification. So you see here that we have a variety of magnetic beads that's available with GenScript. So we have protein A, we have protein G, we have protein AG, protein L, and MMAC protein A beads. Now let me talk to you about MMAC protein A beads, which are the beads which can be reused and which has a binding capacity of about 40 milligram per ml. They are base stable. That means you can just wash them with NaOH and then reuse them up to 40 times. So that's the best part about these beads, that you can just process them inside the instrument and then you can just reuse them again. There are several other beads also which are available, which are mentioned here, and their binding capacities are listed here. We also just recently introduced protein L magnetic beads, and they have a binding capacity of about 15 milligram per ml of antibody or 5 milligram per ml of FAP fragments. Now, let me talk to you about the data that I have for our protein A magnetic beads. So we just compared it with one of our competitors at the response time of our magnetic beads. So you see here that at 0 seconds, both of them are suspended. But at 6 seconds, you see that the MMAC protein A beads, they are all attracted by the magnet, but the one with the supplier B or with the competitor is still in suspension. So you see that the response time of our magnetic beads is really fast. Now, since our beads can be reused several times, we want to make sure that they doesn't lose their binding capacity with when we keep on reusing them again and again. So we checked them up to 40 cycles, and what we found was that there was no reduction in the binding capacity of these beads up till 40 cycles. Also, we looked at the binding capacity of the beads with the variable concentration of antibody, and what we found was that the binding capacity of these beads, the dynamic binding capacity of these beads, is pretty good as compared to the competitor which is in the market right now. Also, since we are washing our beads with NaOH, we want to make sure that there is no protein A leakage from the surface of the beads. And for that, we perform this experiment, and we see that there is hardly any protein A leakage from our beads. We also compared the data for the purity and the endotoxin levels if you're using the assay system and if you're using the resin system. And what we found was that using our assay system and using the resin system, there is hardly any difference in the purity. So using the resin system, it's 98.10, and with the assay system, it's about 97.82. Also, if we talk about the endotoxin levels, there is hardly any difference in the endotoxin levels in the resin system and in the assay system. So it's very, very comparable. So mag beads for the tag protein purification are listed here. So this is the menu that we have for the tag proteins. We have nickel charge beads, we have nickel magnetic beads, we have glutathione magnetic beads, we have streptavidin beads, and we have flag magnetic beads. Now here I would like to highlight our Magnetical beads, which are basically are beads which can be reused up to 100 times, which are chelator resistant and which has a binding capacity of about 10 milligram per ml. You can wash them with 1 molar NaOH and they can be reused over 100 times. We also have other beads like nickel charge beads, glutathione beads, streptavidin and flag, and their binding capacities are mentioned here. Now, this is the performance of our AmMag nickel beads. If you see that, these beads actually, they can be used several times, and we just have to wash them with 1 molar NaOH and they can be reused up to 100 cycles. Now, this is the difference between our nickel beads and our AmMag nickel beads. So, of course, their binding capacities are different. Their application is for AmMag nickel beads is basically for low expression of his-tag proteins or for samples which has high chelators. For the nickel charge magnetic beads, they are basically used for normal his-tag protein purification. AmMag nickel beads is having a reagent compatibility of up to 100 millimolar EDTA and 20 millimolar DTT. In the case of nickel charge magnetic beads, it's only 1 millimolar EDTA and DTT. Now, the reusability of AmMag nickel magnetic beads is over 100 times, but please note that is only in the absence of chelators. If there is high concentration of chelators in the samples, you will be able to reuse them only 5 to 6 times. But in case if you do not have chelators, you can reuse them up to 100 times. For the case of nickel charge magnetic beads, you can reuse them 4 to 5 times, but you cannot wash them with NaOH. You can just purify the same protein that you have purified first time using these beads. You cannot wash them with NaOH. Now, these are the solutions that is provided by GenScript to reduce your culture volume in case if you're processing 500 ml or 1 liter or 2 liters of culture, and if you want to reduce them to a 50 ml tube so that you can process your samples inside the AmMag assay system, we have 2 solutions available here. The first one is the AmMag magnetic wand, and the second one is the AmMag magnetic block. So these AmMag wands, they are available in 2 sizes. They are available in large and small size. And basically, we have this magnetic rod and a sleeve, and the sleeve can be washed with NaOH. What you basically have to do is that whenever your cultures are ready, you put your magnetic beads there, you finish the incubation step, and once that incubation step is complete, you just insert the sleeve into this magnetic rod, and then you just rotate it inside your flask, so that to make sure to capture all the beads. Once you capture all the beads, you just take it out of your flask, and then you just take a 50 ml tube with a small amount of a buffer. You just remove the magnetic rod, and then you just dip the sleeve into the 50 ml tube, and all your beads are going to fall inside the 50 ml tube. And once that is there in the 50 ml tube, you can process that inside the AmMag SA Plus system. The same goes for the AmMag magnetic block. You just can stick your block at the bottom of your flask, and then you can remove all the liquid. You can replace the liquid with the small amount of a buffer, and you can just transfer this into a 50 ml tube. And once you have that in the 50 ml tube, you can process that inside the AmMag SA Plus system. That way you can reduce your culture volume from several liters to just 50 ml tube and still use our automated AmMag Plus system. Now, this is a summary of the entire presentation. So the AmMag SA Plus shortened the purification time. It saves you a lot of time, labor, and money. It purifies anywhere from 2 to 50 ml samples, and it can process up to 12 samples at a time. And it takes anywhere between 45 to 48 minutes to process 12 samples. There is no proprietary buffers or consumables that is required when you're running the AmMag SA Plus instrument. We just provide you with the menu. They're all regular buffers, and we can provide the menu if you need one from us. There's a variety of magnetic beads that's available with GenScript, both for antibody and PAC protein purification. AmMag protein A magnetic beads are alkaline tolerant, and they can be washed with 0.1 molar NaOH, and then they can be reused. They can be reused up to 40 times. They have a binding capacity of about 40 milligram per ml. Our AmMag nickel magnetic beads are also alkaline tolerant, and they can be washed with 1 molar NaOH, and they can be reused up to 100 times. They have a binding capacity of about 10 milligram per ml, and they are resistant to chelators, that is to like EDTA and DTT. There are several other chelators, but these are the ones that I have mentioned, but there's a long list of chelators that they are actually resistant to. We have a variety of solutions that is available to reduce the volume of your samples, like 1 liter or 500 ml, 250 ml like magnetic wands and block that I just talked about. And you can process your samples in MagSA after reducing the size of your samples. Thank you so much for your time. And if you have any questions, please feel free to email me at nishant.saxena@genscript.com. And in case if you are interested in the demo, we are doing free demos of the instrument. We can bring it to your site and we can give you free beads to process your samples. And in case if you are interested in that, please contact us at product@genscript.com. Thank you so much.